All right, guys, so this is section 3 of our genetics unit. This is going to be really quick, kind of the same principles that we have talked about thus far. So we're just going to go through incomplete dominance or non-dominance. And section 4 will be codominance, and then we'll go on to pedigree charts from there. Okay, so section 4 and 5, codominance and pedigree charts will be a little bit longer, but I'll try to keep them as short as possible. Okay, so let's go ahead and get started with, I think there's 4 or 5 slides is all here, so let's knock these out and then I'll talk to you about the worksheet, okay? Really quickly, I want to finish up with where we ended last PowerPoint, and that was this slide, and it says this is B, a summary of Mendel's principles, letter B. It says there are some exceptions to the principles, and specifically the principles that it is talking about is independent assortment, and we talked about dihybrid crosses, monohybrid crosses, and dominance and recessiveness. So there's some exceptions to these principles because not all genes show dominance or recessiveness. For some genes, there are more than 2 alleles, and then many times these traits are controlled by more than one gene. So now we're going to start talking a little bit, so non-dominance, incomplete dominance, codominance, sex-linked traits. So we're going to talk a little bit about the exceptions of Mendel's rules. And this is where genetics gets a little bit more tedious, not necessarily with incomplete dominance or non-dominance, but definitely with codominance, okay? All right, so genes in the environment. So let's just put this up here and talk about these real quick. Again, this is pretty quick. This is number 9 on page 11. A, gene expression is always the result of the interaction of genetic potential with the environment. Obviously, the environment plays a role in our development. And for instance, it says a seedling, right here, may have the genetic capacity to be green, to flower, and to fruit. But it will never do these things if it is kept in the dark. A tree may never grow tall if the soil is poor and no water is available. So the expression of the gene relies heavily on the environment and the correct environmental factors. If we're lacking water, for instance, for a seed, if we're lacking sunlight, poor soil, then those genes are never going to come, are never going to be expressed, and therefore we'll never see the phenotype that they're coded for, simply because the environment did not interact appropriately with the genes, okay? So in other words, the presence of the gene is not all that is required for the expression of a trait. The gene must be present along with the proper environmental conditions. The phenotype of any organism is the result of interaction between genes and the environment. A reminder, phenotype is the characteristics or traits of an organism. All right, so real quickly, let's knock out incomplete dominance and non-dominance. This is number 10 at the bottom of page 11. This is pretty simple. Same concepts as monohybrid and dihybrid Punnett squares, but we do have some incomplete dominance here, okay? And I'll explain what that is. So all traits are not clearly cut as dominant or recessive. Some are what we call non-dominant or blend together to form incomplete dominance. So for instance, here you have a red flower or white flower, and instead of making a red or a white flower, they make a pink flower. Okay? So for example, in some flowers such as snapdragons and 4 o'clocks, a homozygous, remember that's 2 of the same allele, crossed with a homozygous, so a homozygous red flower crossed with a homozygous white flower. So you could think of that as like big R, little r, yields a heterozygous pink flower. And this is known as incomplete dominance or non-dominance because no allele is dominant or recessive. They're, you could think in a sense, equal. Okay? So they blend together in the offspring. Since there's no recessive allele, use only capital letters. For example, a red flower would be big R, a white flower would be big W, and the pink hybrid would be RW. So this is a little bit different than what we've done before, so you can kind of ignore what I said earlier about big R, little r. you're going to have what looks like two alleles, simply because they are not dominant recessive, they're codominant. So red flowers have one allele, white flowers have one allele, and together the combination makes a different color or phenotype of the flower. So what type of offspring may be produced by two pink flowering plants? So let's look at this real quick. Genotypes of the parents, remember these were heterozygous, it said, so big R, big W. So if we cross these, this is a monohybrid Punnett squared, just like we're used to. We're going to have homozygous red, heterozygous, and then homozygous for white. Okay, so we're going to have one red flower, one white flower, and two pink flowers. Okay, there you go. Pretty simple. All right, in a certain plant, flower color shows non-dominance, but stem length shows dominance. Okay, so let's think about this. Flower color shows non-dominance, but stem length shows dominance. The allele for long stem is dominant over the allele for short stem. Cross a heterozygous, long-stemmed red plant with a short-stemmed pink plant. So remember, this is a little bit different because the stem length is dominant. So we're going to use, you could use big L, big R, big little r. But for the pink and red colors, we're going to use just two large capital letters. Okay, so let's look at this real quick. Genotype of the first parent. So I don't know what actual genotype or letter they're going to use, but it's going to be, remember what I told you, since these are dominant recessive, you have to use big R, little r. Okay, and then big R codes for a red flower. Remember, this is shows non-dominance or incomplete dominance. And then the second parent, remember it said heterozygous long stem. So if it's heterozygous, it's going to be, let me see here. The long stem is dominant. Okay, so this is the heterozygous long stemmed red plant. Now we're going to do short stemmed pink plant. Remember, short, so long stem is dominant over short stem, so we're going to have two little r's, and then we're going to have a pink plant, so we're going to have big R, big W. Okay, so try to wrap your brain around this a little bit. If you have any questions, let me know. The idea here is that the first two alleles show dominance and recessiveness for stem length, but the last two alleles, they're capital letters because they're incomplete or non-dominant. Okay, so same thing. We do the same exact thing here that we always would. Big L, big R, big L, big R, little L, big R, little L, big R. There you go. And then we divide these out the same exact way as we had before in the last section. So here we go. Then we combine all these. You should know how to do this by now if you did the second homework. And then all we do, guys, is we count up the genotypes and the phenotypes. So let's go over these real quick. This is going to, anything with a one big L, one capital L, is going to be long stem. So long stem, 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 long stem. And all of these are going to be short stem, two recessive alleles. So half are long stem, half are short stem. Now, anything with two R's is red. Anything with RW is pink. And then there's no WWs here. So we have half long red. Okay, so let's think about this. I'm sorry, we have four long red, four short red, four long pink, four short pink. Okay, so 16. So wrap your brain around that a little bit. Just exactly what I just said, okay? So any questions about this, just let me know. All right, so we're not going to go on to codominance right now. That's a little tricky. We'll talk about that next, but I do want to talk to you about the worksheet here, okay? So let's scroll down a little bit and go over a couple of these really quickly. I'm not going to do all of them for you. I'm probably going to do one of each. And let me fix this real quick where we can see everything here. All right, so let's go over this first little bit here. I'm going to put this where I can draw on the screen. Whoops. All right. So I should be able to draw now. So let's read this. In chickens, BB calls for black chicken. WW calls for a white chicken. BW combination calls for a blue Andalusian chicken. Cross a blue hen. So we have blue, which is BW, with cross a blue hen with a blue rooster. So two BWs. So BW, BW, big B, big B, big B, W, B, W, W, W. All right, so let me mark these out because I shouldn't have drawn that there. Our genotypes are B, 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 W, and W, W. And there's one of these, two of these, one of these. So we're going to have one black, whoa, sorry about that. Two, let's say blue, and one white. Again, this is called non-dominance. Okay, so there's that one. Now we're going to do a black chicken. So that's big B is mixed with a Andalusian blue chicken. So heterozygous. Then we come up with this. Oh, I messed up. Sorry. Let me see if I can undo that. There we go. All right. All right, so we have genotypes are B's and BWs, half of each. So two black, two blue. All right. Let me go scroll down here. So if there's any questions about these, just let me know, okay? All right, so let's do a dihybrid cross now. Chickens, rose comb is dominant over single comb. So remember, this is dominant. This is the big thing. So we know we're going to have a dominant allele and a recessive allele. What types of offspring would be expected from a white? Remember, color. So comb is dominant or recessive. White, though, is non-dominant. Okay, so this would be BW from above, and this would be WW. So now we got to think about this. So what types of offspring would be expected from a white rooster who is heterozygous, rose-combed? So we would have rose-comb heterozygous would be big R, little r, but it's white, so we're going to have W, W. With a single combed, so it's single combed, so it has to have two recessive alleles because it's a recessive trait, and it is blue. So little r, BW. Okay, so let's separate these. R, W, R, W, little r, W, little r, W. Then we got to go here. So little r, B, little r, W, little r, B, little r, W. Okay, So I'm going to do this one for you. The rest of them, I'm just going to show you do the genotypes for you, and then you can work the Punnett squares yourself. But here we're going to go big R, little r, big R, little r, big R, little r, big R, little r. So we know all of those, actually all of these too, are going to have a heterozygous rose comb. So they're going to be heterozygous for a rose comb. Okay, these are going to be recessive, so they're going to have single combs. And if you want to know what single and rose comb looks like on a chicken, just Google it. It's a real thing. Okay. Then we're going to have oh, I messed up. These are W's. And these are W's. I got in the rhythm of writing BW. Okay, so these are white chickens, white chickens. So B, W, W, W. Messed up again. So make sure you pay attention when you do these. It's easy to mess up, okay? All right, so let's write the genotypes. I have heterozygous, rose comb, BW. And how many of those do I have? So 1, 2, 3, 4. Then I have still heterozygous, but I have white chicken. And I have 1, 2, 3, 4. Then I have homozygous recessive, and this is going to be a pink, or I'm sorry, a blue chicken. So 4. And then last but not least, so 4 of those. So I've got, let's do this. Let's do rose, and I have 4 rose blues. I have 4 rose whites. We have single, and blue. And single, and white. Okay. All right. So from now on, I'm just going to give you the genotypes, and you have to do all of this. Okay. All right. So let's go through these one at a time and see what we got. Okay. All right. 4 o'clock flowers. Flower colors due to non-dominance. Some red flowers, some are red, some are white, others are pink. Cross a red flower with a pink flower. So what is the genotype for a red flower? We can just make this up, big R. Since it's non-dominant, they're both capital letters. And then what is the genotype for a pink flower? Well, it gave us this earlier, so we'll do RW. All right, then you guys need to do the rest of this. So I gave you the genotypes. So you'll fill this out. Do the Punnett square, write the genotypes, phenotypes, and then do these probabilities, okay? It's actually pretty easy. All right, let's look here in cattle. RR genotype calls for a red cow. WW calls for a white cow. RW calls for a roan color. So a red bull. So we'll do red. Cross with a white cow. And then you do the rest, okay? So I gave you those. So red, white, and you're good to go, okay? So let's go on to 6. Well, okay, let's go to 6. Cattle breeder knows that hornless condition is dominant. So look here, we're going to have a dominant recessiveness. He mates a heterozygous hornless cow who is roan colored. So that's from above. We did, what did we do? RW before? I think we did, let me check. We did RW, okay. So RW are our colors here. Remember so we did a heterozygous hornless cow. So hornless is H, it's heterozygous. So hornless is the dominant trait. H, H. That's roan colored. So R, well, sorry. So we did a heterozygous hornless cow with a roan color that was roan colored with a horned roan bull. So horned is recessive, and roan would be, I'm sorry, yeah, roan. So you take these genotypes, fill them out here and here. So these go here, and then you do the rest, okay? I think you can do that. All right. Shape and colors of radishes are controlled by 2 independent pairs of genes that show non-dominance. The color may be red, white, or purple. What offspring might be expected when purple radishes are mated with white radishes? I'm not sure why that looks like that. Let me fix this real quick. Okay, there we go. All right. So we have what offspring might be expected when purple radishes, so RW, are mated with white radishes. WW. You do the rest, okay? Then this says the shapes, colors of radishes are controlled by two independent pairs of genes. It says that above. We already read this. The shape may be long, round, or oval. So look, now we have two genes, two pairs of genes that both show non-dominance, okay? So show a cross between a long red and round, a long red radish. So long is LL and red. So I'm going to do R, red, or long is LL, okay? And then, so that's this. And then a red radish, a long red radish, and a round white radish. So white is W, and round is O. So that's this. Okay. So genotype, we've already wrote that, so I can do this for you. Long red radish is R, L. Round white radish is W, O. Now you'll have to do a dihybrid Punnett square for these. So do that on the back or something if you want to, and then you can get this. Okay. Now let's see what this next question says. Let me get out of this real quick. Scroll down. So you'll have to do the F1 generation. Okay. So I'm assuming all of the F1 generation. Well, let's look here. Make sure I'm telling you right. So I told you earlier that we did a R, L, with a W, O. So if you cross those, I mean, you would have R, L, R, L, R, L, R, L. And then over here you would have W, O. So this is exactly what I expected. So all genotypes will be, if I did this, will be R, W, L, O. All of them will be that way. So all genotypes will be R, W, L, O. So we're going to have purple, so purple and oval. All right, so now it's saying let the F1 generation self-pollinate. So we're letting this self-pollinate. So we're going to have R, your genotypes for this Punnett square below will be RW, L, O times R, W, L, O. So just put these here. Same thing. So it'll be RL, RO, WL, WO, RL, RO, WL, WO. Do your genotypes and phenotypes. Okay? Pretty simple. Let's see what else we have. Okay, so we have 9 and 10. So let's knock these out real quick, see what we got here. All right, so in some cats, the gene for tail length shows incomplete dominance, okay? When a cat has a long tail, when a cat with a long tail is bred to a cat with no tail, the resulting offspring has short tail. So we have long tail, no tail, short tail. So predicted, so a cat is bred long tail cat, so we'll do L is bred to a cat with no tail. So we'll just do N. And then you'll cross those. The short tail cats will be LN. Okay? So do your genotypes, phenotypes. Then we have a long tail cat, so that's LL. Bred with a short tail, or with a short tail cat, so LN. And then down here we have 2 short tail cats, so it's LN. I don't know what happened there. So do your crosses, phenotypes, and genotypes. Pretty easy. Pretty easy. All right. So let's do this last one, and you are done with section 3. All right, so in a particular plant, long stem is dominant over short stem. I already said that. Let me fix this real quick. Long stem dominant over short stem. So we'll just call this L, and short stem will have to obviously be 2 recessive alleles. Same plant may produce flowers of red or white. So this is R, and then W. Across a heterozygous long stem, so long stem heterozygous is L with a pink plant. So that's the hybrid, so to speak, RW. So when red's mixed with white and we get RW, that it's non-dominant, so it's pink. Okay? And then we're going to do a heterozygous long-stemmed red. So L red. So you cross those, do this, do that, and you, my friend, are done with section 3. Okay, remember, test on Friday. Have a good day.